Okay. Okay, I am not, in fact, Terry Sanofsky, but in his absence, it gives me great pleasure to welcome all of you here this afternoon. I am pleased to introduce our visiting speaker, Mark Iacoboni, who comes to us from UCLA, where he is the director of the Transcranial Magnetic Stimulation Lab. And most of you already know a little bit about the topic under discussion, which has to do with mirror neurons. And as you know, Mark has been a prominent researcher in exploring mirror neurons and their scope and limits. I will also draw to your attention a book that he has authored called Mirroring People. And this, I guess, is a mirrored person here. The book is not officially out yet, even though it is physically out and compiled, but it will be out later on towards the end of May. So thank you so much for coming, and we're all looking forward to your talk. Thank you.
Thank you for your lovely introduction and also for showing the book. It's always good for selling copies. Okay, so what I'm going to do, first of all, I'm going to stop my crowd. I'm going to give you a little overview of the kind of things I want to discuss today. And I want to start really talking about the mirror neuron system in monkeys, the basic findings, and also try to explain some aspects of this system that I don't think that a lot of people grasp. Then I want to say a few things about what we have done in the human mirror neuron system, mostly using brain imaging. And these, I'm going to most of my talks are actually centered on brain imaging. But today I'm going to present with some detail data that we just finished collecting. We have a paper it's under review right now. And so that will be the third part of the talk. And here I'm going to present human single unit data that show that human neurons have mirroring properties. And the reason why I call this pervasive mirroring is that because it's not just, oh, we found these cells also in the human brain, but there's plenty of new findings that we have discovered with these recordings in the human brain. Okay, let's start with the monkey system. And let me just remind you a very basic concept. Rizzolatti and collaborators weren't really looking for these kind of cells. They were studying motor control. And they were especially interested in grasping, which is a fundamental act of your life. Try to get out of the house without grasping the doorknob. You grasp things all the time. While in the primate brain, there are two major cortical centers for the control of grasping. One is located in area AIP, which is a little tiny area in the anterior part of the intraparietal sulcus. And the other one is located in the ventral premotor cortex in an area called area F5. So they were studying grasping, and then what happened was that they found that some of these cells had properties that were not expected. There's a lot of, the media love mirror neurons, and so there's plenty of stories about the way these cells were found. And I think I want to clarify this. It wasn't just a sudden recognition. The idea that premotor cells could fire at the site of the action of somebody else was something that none of those researchers could even conceive. So it took them almost years to really grasp the concept that the cells were actually firing at the site of somebody else's action. It's not something that, oh, they grabbed the peanut and they found about it. I'm going to show you some clips that show the properties of these cells, the basic ones. You're going to see a monkey grasping 3 times, and every time the monkey grasps, you're going to hear the discharge of the cell. Those are action potentials of the cell fire when the monkey grasps. So far, so good, because an electrode is located in the premotor cortex, and those cells are supposed to fire when the monkey makes an action. Now, the same cell, you're going to see the next clip, is going to fire when the experimenter in front of the monkey grasps something, even though the monkey is completely still. A real monkey see, monkey do cell. My colleague Isaac Fried, when we watched these clips, he said, this neuron has not read the handbook on neurophysiology. He's not supposed to fire. In the next one, I'm going to show you another visual property of the same unit. In this case, the cell also fires in response to the sight of Leo Fogassis bringing something to the mouth. So the visual properties of these cells are often broader than the motor one. Not necessarily. Some cells are called strictly congruent mirror neurons, and they fire only for the same action. But it's important to grasp the concept that, in fact, the visual property of these cells, in most cases, actually respond to multiple actions. And that helps when it comes to interactions with other people, because with mirroring only, you don't get too much mileage. You have to also interact with others, do complementary actions, and so on. Fogassi is very pleased because he's Italian, so anything has to do with food. In the next one, I'm going to show you what Leo does is to get a syringe and put the syringe in the mouth, and you hear the discharge of the cell. Why? Because the monkeys that live in the lab get juice through a syringe, and they love juice, and so for them, it's really a useful tool. But then you're going to see Leo doing the same action again, except that the last second he makes the trajectory of the hand going outside the mouth, and you're going to hear no discharge. That means that the cell really needs to see the whole action in order to fire. And the last one, it's not going to show you anything that is really novel because the basic property has been shown by these other clips. But I think it captures something that is really interesting about these cells. You're going to see Leo grasping something, putting it in the mouth, and then offering the same piece of food to the monkey. And the cell fires both when Leo puts the food in his mouth and both when Leo offers the food to the monkey. And to me, it captures visually really the major concept of these cells, which is about sharing. All right, these are compelling videos, but you don't publish this stuff in scientific papers. What you publish are these kinds of data. These are on the top. This is called raster. It shows each line is a trial, and every mark is one action potential. And you see that for each trial, there is an increase in the fire rate for when the monkey grasps the object or the piece of food, but also when the monkey sees it. And this is called histogram, and plots the fire rate change over time. And one thing you can see is that there is, of course, a discharge for both execution and observation of action, although it's slightly larger for execution. And that's an important concept also to remember. So they found these properties, and what the idea was that, well, this seems a very simple mechanism that evolution has devised to make us understand in a very immediate way the actions of other people, which is a fundamental function when it comes to social interaction. You want to understand what other people are doing. And so the idea was, how deep can these cells go? And one thing that they found was, very early on, it was found that, in fact, as I told you, the cell fires only to the complete action. For instance, pantomimes do not trigger the activity of the cell. So I'm going to show you first a grasping action. The cell fires, but then same thing except that there is no object to be grasped. It's only a pantomime. There is a much weaker discharge. Now, what happens if the monkey sees that there is an object here on the table, and then there is a screen that slides in and covers the sight of the object, and then the human experimenter reaches behind the screen? The question is the mirror neuron going to fire? So the cell in this case fires even though the completion of the action is not observed. And the control condition is a starting point in which there is no object, the screen comes in, and then again the human experimenter goes behind the screen. This is not even a good trial to show, but it's a much weaker discharge, and I'm going to convince you with the rasters and histograms of the original paper. This was published in Neurons, and the title of the study was "I Know What You're Doing." And so the idea is that the baseline conditions, you see a grasp in action, there is an increased firing rate, you see the pantomime, there isn't. And then the two hidden conditions, in which there is a screen, strong change in fire rate when the monkey knows that there is an object behind the screen, and practically nothing when the monkey knows that there is no graspable object behind the screen. Now, keep in mind that these two conditions at this point are visually identical. The difference is that the monkey knows the prior knowledge of what's behind the screen. So, in fact, initially when the Parma people found these cells, they thought, haha, these are intention cells. They really code for the intention. But then, a more conservative hypothesis is that what they actually do is, again, based on prior information, they know that one hidden action is a grasp and the other hidden action is not a grasp, and that's the kind of coding they make. But still, it is quite amazing that they even respond to hidden actions. Now, actions, sometimes they also make sound. So the question was, do these cells fire in absence of the site of an action, but just while listening to a sound of an action? And what I'm going to show you next is a little clip in which you're going to first see a static picture, and then you're going to hear the sound of breaking a peanut, and then you're going to hear the discharge of the cell. Then you're going to see the silent clip of somebody breaking the peanut, and the cell discharges again. And finally, in the last segment of the clip, you're going to see and hear the action and the sound of breaking a peanut, and again, the cell fires. And again, these are the histograms of the original paper that show robust firing rate changes for vision and sound of an action, but also for the sound only, and no changes in firing rate for control sounds. So these cells fire when the monkey makes an action, when the monkey sees somebody else making the action, when the monkey sees a hidden action, but based on prior information, knows that there is something to be grasped behind the screen, and also they fire to the sound of an action. They seem to have a fairly abstract coding of the actions of other people. And so far I've been showing you only grasping mirror neurons, but in fact, and I think this is very important when it comes to understanding the emotions of others, there are also mirror neurons for facial expression and for mouth movements. And this was a study that was published in 2003, and the lead author was Pier Francesco Ferrari. He's an interesting fellow. He got a PhD in studying monkey behavior with Elisabetta Wieselberghi, and then did a postdoctoral fellowship with Rizzolatti doing neurophysiology. He has a very unique background. And you can tell that he studied monkeys' behavior because he's pretty good at doing lip smacking. So the two main categories of actions were ingestive actions and communicative action. Again, this lip smacking, which is a facial gesture of positive valence in the monkey world. And there are mirror neurons for both types of action. This is a cell that fires when the experimenter bites the banana and when the monkey also bites the banana. This is another unit that fires for when the monkey sucks from the syringe and when the experimenter makes this lip smacking action. And also a full communicative mirror neuron, there's plenty actually, that fire for both the execution and the observation of lip smacking. Again, this is very important because it gives us a connection to understanding in this kind of simulation mode the facial expressions of other people. Now, the original findings of these cells were recorded, as I told you, in area F5. When Rizzolatti and collaborators found these cells, they also knew that there was another lab in Europe, the lab of Dave Perret, and they were studying higher order visual cells in the superior temporal sulcus. And the visual properties of the cells that were studied by Perret were very similar to the visual properties of mirror neurons. So, of course, for Rizzolatti and collaborators, it made sense to think that the visual information was conveyed from STS to F5. On the other hand, these two regions in the monkey brain are not anatomically connected. So they needed a third region that would connect these two ones. And there is one, in fact, area PF in the rostral part of the inferior parietal lobule that connects both with STS and with F5. So it made sense to think that visual information was relayed from STS to F5 through PF. And just based on this reasoning, Rizzolatti and collaborators decided, let's make also recordings in this PF area. Maybe there are mirror cells also there. And in fact, they found mirror cells also in PF. So what we know now is that there are two major areas in the monkey brain that contain mirror neurons. Doesn't mean that there aren't other brain regions that contain mirror neurons, but what we know for sure is that these two have mirror neurons, area F5 in the ventral premotor cortex, and area PF in the rostral part of the inferior parietal lobule. Now, this circuit between F5 and PF, it's embedded in a very large parallel architecture connecting parietal regions and frontal regions. The frontal, on this table, the frontal regions are here, and the parietal ones are here. Why I'm making this point, I think it's important to consider where this circuit is located, because all these circuitries, all these connections, they're all dedicated to one major goal, which is sensory motor transformation, transforming sensory input into an appropriate motor output. And if you keep that in mind, then you have to really consider that in evolutionary terms, that's exactly what happens also with mirror neurons. What they try to do is to transform a visual input into a motor output. But what kind of visual input? Of course, we're talking about actions. So what kind of sensory motor transformation we're talking about here? Well, I would say imitation is a fundamental sensory motor transformation that helps you understanding the actions of others and copying them. And in the human brain, one thing we know in humans is that we are really good at imitating, and we start very early on. This is the work of Andy Meltzer that shows that even newborns can imitate at least some rudimentary facial expressions. I always tell Andy when I see him, with your work, it was an amazing work. It changed the way we thought about imitation, but you also accomplished something that nobody else will do. That is, he managed to publish a paper in Science with a picture of himself sticking his tongue out. So we thought, well, at some point when the discovery was made in the 90s, in the mid-90s, Rizzolatti and collaborators wanted to do some brain imaging, and so we wrote a grant together, a Human Frontier grant, because I was already in the States. By the way, a lot of people think that I'm from Parma, because I'm Italian, I do mirror neurons, I collaborate with Parma people. It's not true, I'm from Rome. So we thought, well, let's try to do some imitation paradigms to see whether or not there are brain areas with similar mirroring properties. But at that time, now there are some data on spiking activity in single cells and bald fMRI. At that time, there was nothing. So we thought, let's use the most parsimonious model, because I think that's what science should always do. When you have nothing, when you start a project, you want to start with the simplest possible explanation. Only when your simplest model failed to accommodate the data, you want to come up with more complex models. And so we started from the data from the monkey, and we noticed again that there was a much more robust discharge for making the action than observing it. And so we conceptualized something similar in terms of bald fMRI, such that there is a bigger bald response for the motor task than for the action observation task. But also we thought, well, during imitation, you're doing it and you're seeing it at the same time. So we thought, well, maybe there is additivity of these two signals, and so imitation should have the higher signal. And that's exactly what we predicted as mirror neuron activity in areas of the human brain that had these kind of cells. We did a very simple experiment in which subjects were just watching and imitating finger movements, and they had some control conditions. And we found 2 brain regions that had this profile, and they were anatomically close to the monkey brain regions that contain mirror neurons. One is in the inferior frontal gyrus, and the other one is in the rostral part of the posterior parietal cortex. So far, so good. We found this system, and it seemed very similar, even anatomically, to the monkey brain system, mirror neuron system. But of course, we had a little problem. Well, actually, before telling you what kind of other experiments we did, let me give you, since then, in the last 10 years, we've done a lot of studies on imitation and using brain imaging. And rather than going into the details of the studies, I want to just give you an overview of what we think are the most important brain areas for imitation. You need the whole brain to imitate. You need also B1, because if you don't see, you're not going to be able to imitate. But these 3 regions that are very similar in terms of their functional properties and their anatomy to the monkey brain regions that are important for action recognition and for mirroring, I think they're really what we call the core imitation circuit. And of course, it starts from the posterior part of the superior temporal sulcus, which provides a visual pictorial description of the action that you're going to imitate. And then the parietal component of the mirror neuron system would be concerned more with the motoric detail of the action to be imitated. And finally, the frontal component, which overlaps with a famous language area, Broca's area, is concerned more with the goal of the action. I reviewed lots of papers in which people say that the mirror neuron system in the human brain is composed by STS, this region and that region. And that's wrong, because, at least it was wrong until a few months ago, because these areas, as far as we know, do not have motor properties, so they cannot be considered mirror cells, because you need to have discharge for both the execution and the observation of the action. But, brain research gives you surprises. And what John Brugman says, when data make you change your mind, that's science. And I changed my mind, and I'll show you that later. And these areas, of course, are connected, and there is a whole functional circuitry that we have studied over the last 10 years. Now, this is also important because there is a hypothesis that was proposed by Rizzolatti and Arbib in 1998 in a paper entitled Language Within Our Grasps that suggests that mirror neurons are really important neural elements for evolution of language. And one of the arguments is an anatomical one. And the idea is that area F5, in which mirror neurons are contained, is similar, homologous to Brodmann area 44 in the human brain. And I'll try to show you this visually. So this is, again, the drawing of a monkey brain, and you see the lateral wall of the brain. The frontal lobe, this is the central sulcus, and this is the frontal lobe. The prefrontal cortex is up here in the front, and the premotor cortex is behind this sulcus. This is called the arcuate sulcus because it's like an arc. And it's composed of 2 sectors. The blue one, it's the dorsal sector, and the red one is the ventral sector. Now, if you just look at the sulcal pattern, it turns out that it appears that what happens during evolution is that the arcuate sulcus opened up, and it became 2 parallel sulci. That in the human brain are the superior frontal sulcus and the inferior frontal sulcus. Now, if you look at where if you believe this story, and if you look at where F5 is located with respect to the ventral part of the arcuate sulcus, it's clear that it would be pushed down in the posterior part of the inferior frontal gyrus, exactly where Brodmann area 44 is located. There are also other anatomical arguments in favor of this homology between area F5 and Brodmann area 44. So when we actually had our brain imaging studies showing that pattern of activity in Broca's area, that was an important empirical evidence in support of this evolutionary story. And also we had done some studies showing, in this case what we were doing is to combine brain imaging data with post-mortem data that show the probabilistic distribution of Brodmann area 44 in the human brain. And in fact, our peaks of activation, this is a meta-analysis of 58 subjects that we studied in my lab, show that the peaks of activation overlapped entirely with the red blob, which corresponds in cytoarchitectonic terms with Brodmann area 44. So the story is compelling, but there is always a little caveat. Because when you do brain imaging, getting activation in a language area is always a double-edged sword. Some people started saying, how do you know that these activations are not just covert verbalization of the task? It didn't really make a lot of sense because we predict a whole pattern of activity, and covert verbalization would predict similar pattern of activity throughout all the tasks. But we thought, well, we have various tools in cognitive neuroscience these days. Why don't we use another one that gives, rather than giving you correlational information as brain imaging, gives you causal information. So we used TMS, and we thought, let's zap Broca's area, and let's see what happens. So this is going from brain mapping to brain zapping. So we did an experiment in which subjects were doing an imitation task and a control task, in which they were also required to make the same motor output, but in this case in response to a symbolic cue. And in my lab, we have this system it's called frameless stereotaxy. We can visualize the brain of our subjects, not removing the skull, because that's not allowed, but using the virtual images collected in the MRI lab. And so we can see exactly where we stimulate. We stimulated, of course, over Broca's area and also over a control site. So we had a control task and a control site. And what we found was that only when we were stimulating zapping Broca's area and the subjects were doing the imitation task, there was a reliable increase of errors. So there was a selectivity, a specificity such that only Broca's area and not the control site, and only the imitation task but not the control task, were affected by TMS. So this, to us, suggested that in fact Broca's area is an essential area in imitation, and the activity that you see in brain imaging studies, it's not an epiphenomenon due to covert verbalization. And so this is, again, a picture of the whole system. The two red areas are the human mirror neuron areas, and the yellow one is the visual input to the system. So far, so good. However, none of these data in the human brain go to the resolution of a single cell. And something amazing, I think, happened in the last 2 or 3 years. Some people started saying, well, we don't really know if mirror neurons are in the human brain, in spite of all these data and evidence from the imaging work that we have done. It is compatible with the idea that mirror neurons are in the human brain. Now, this is really, to me, it's an astonishing proposition because, in fact, a common practice in neuroscience is that you get the cellular mechanism with neurophysiological studies in the monkeys, you move in the human brain using brain imaging techniques, and when you see that there is a nice correspondence in terms of the behavior of a neural system level in brain imaging, the behavior at cellular level in the monkey neurophysiology data, you make the inference that is a very reasonable one, that the same cells that are in the monkey brain are in the human brain. Just to give you a couple of examples, we know that in the dorsal premotor cortex is very important for conditional motor learning, that is the ability to emit a motor response to an arbitrary sensory stimulus. And you do that all the time when you brake, when you're driving and you use the brake in front of a red light. That's an arbitrary association between a visual stimulus and a motor response. And there's plenty of single unit data that show this kind of behavior at cellular level in the monkey brain, and also brain imaging data that show the same thing in the dorsal premotor cortex of the human brain. Nobody's ever said, well, we don't really know if those cells in the monkey brain are also in the human brain. Another beautiful example of going from the monkey neurophysiology to brain imaging, the reward system, striatum, the cell, the firing that predicts the expectation of reward. The same thing has been modeled in brain imaging data. It's been shown with beautiful experiments. And as far as I know, nobody ever raised the issue, well, we don't really know if those cells that Wolfram Schultz found in the monkey brain are also in the human striatum. But when it comes to mirror neurons, this amazing statement was made. And I think that what I'm going to show you next, which is the single unit data that show mirroring properties in the human brain, is not a response to this, because this statement is so silly. But in a way, it's also a bit dangerous. We live in a country in which the evolutionary framework is actually challenged for no reason, because the scientific facts are really so much in support of it. And also we live in a state in this country in which animal rights activists are so aggressive that to make statements of this kind that almost undermine the logic of doing experiments in animals, to me it's just dangerous. Anyway, let's move into the data that we collected. So this is an amazing and rare clinical opportunity. There are patients that have to undergo surgery, brain surgery. They have epilepsy that is not controlled by drugs, and the surgeon needs to find out exactly where the epileptic focus is. And to do that, they implant electrodes in the brain of these patients. And of course, this is a very invasive procedure, and the only reason you do this is for a medical reason, not for scientific curiosity. So we have no say where the electrodes were located, are going to be implanted, and we have no say for how long they get implanted. The way the surgeon does the procedure is that they implant the electrodes, they take away the drugs from the patient, and they wait until the patient ceases. When the patient ceases, they have very good data that tell them exactly where the focus of epilepsy is. Now, during this time, you have the unique opportunity of doing experiments, if the patients allow, of course. And so my good friend Giacomo had done some studies in the 90s on totally different issues. He's been doing this for a while, and at some point I told him, well, why don't we do something on mirror neurons? And initially the whole thing almost died right away, because he told me, well, but where are mirror neurons located? Well, they're located in area F5, and we believe that in the human brain they're located in the ventral premotor cortex. He told me, Marco, we're going to go nowhere here, because the way we implant the electrodes, we are not going to record in the areas that you think have mirror neurons. The whole thing died, and we were busy doing our own things, until at some point, this fellow, which is a postdoctoral fellow in Itzhak lab, Arna Ekstrom, and he's interested in aspects of visual spatial memory. And he has a task in virtual reality according to which the patient drives a cab, and he's supposed to pick up various people in various places. And of course, it's a computer kind of game. And what Arna noticed was that one of the cells of his patient had a really robust response every time the patient was pressing a key. And so he thought, it's in the frontal lobe, it's got to be a motor cell. He came to me and he told me, look, I got this cell, it's a motor cell, why you and Itzhak are not doing something together? And I told him, well, where did you record that cell? Itzhak told me that you guys don't record from the area that I'm interested in. And he said, well, you're right, it's actually in pre-SMA or SMA. And then I had this sudden realization. Pre-SMA and SMA are homolog of a monkey brain region called area F6, which is anatomically strongly connected with area F5, in which mirror neurons are found. And so I thought, well, says who that mirror cells must be located only in the areas in which Rizzolatti has recorded? And this other fellow came aboard, Roy Mukamel. He's now postdoc in my lab, and he's the leading author in this experiment. I call this pervasive mirroring because we found mirror neurons pretty much everywhere we recorded from. And let's try to conceptualize this. I like to make this quote, when people are free to do as they please, they usually imitate each other. It's a nice quote, it's funny, and it's also true. Imitation is pervasive. On the end, we do not imitate all the time, right? So you get to have some control mechanism in the human brain in order to control this tendency to imitate others all the time if you have these mirror cells. And so I've been thinking, well, let's go back again to the drawing board, and what these are the original findings from Gallese. You see that there is this much more robust discharge for the execution rather than the observation. This suggests that, in fact, this weaker response during observation allows not to imitate the actions that you're seeing, such that in order to actually execute the action from the firing of this cell, you need to reach a threshold that it's reached only with much more robust discharges. But of course, this just changes the question. Then how does the cell know not to fire as strongly as this one? And again, if you look at the monkey brain, this area in which we know mirror neurons are located is strongly connected with area F6, which is the homolog of pre-SMA in the human brain. And through indirect connections, it's also connected to some cingulate motor areas, So I thought, well, maybe what Arna and Itzhak are doing, they're recording from areas that control our mirror neurons in the ventral premotor cortex. So it makes sense to think that maybe there are mirror neurons also in those regions. The way the experiment is done, this part of the drawing, forget these little things here, but this part of the drawing is the classical electrode that is used in medical settings. The dark bands are the bands that record EEG signal. The electrodes are implanted this way, and they reach the medial wall of the two hemispheres. Now, what Itzhak did was a very smart modification of this basic setup from which you can get only EEG signal. He put some microwires on the tip of the electrodes, and there are 9 microwires for each macro electrode. One is the ground, and the other 8 microwires, they record spikes and waveforms from single and multi-unit. So you have the opportunity to record a single neuron activity in the human brain. And the first experiment we did, it took us 3 years to do this experiment because, these patients don't come every week. And sometimes you can't really use them, and we have only half an hour of time, basically, because you can imagine how many experiments are done on these patients. But so the idea was to do a very simple experiment in which subjects are watching either a word on the screen. In this case, finger means that the patient is supposed to do a precision grip, like when you grasp the handle of the cup. And when they see the word hand, they do a whole hand grasp. They grasp the body of the cup. And then they do also facial expressions, in this case, smile or frown for the execution part of the task. And then the observation part of the task, they watch these grasping actions or they watch facial expression. We also have control conditions in which we show the words without, and we ask the patients not to do anything, just to check whether or not the same cell fires for just watching the command. So these are the basic conditions, just the idea of looking into mirror neurons for grasping and mirror neurons for facial expression. And these are some rasters and histograms of cells that have mirroring properties in the human brain. You see the activity, the increased firing rate during the execution of a facial expression and also during its observation. And we have a variety of these cells. I'm going to give you the numbers in a later slide. Some cells increase the fire rate and some other decrease the fire rate for both the execution and the observation. And these very same cells do not fire for the control conditions, of course. But we also found what I call super mirror neurons. There's been a whole debate. How do we call these cells? Well, in the paper that we sent out a few weeks ago, we don't call them in any way because we don't want to trigger any psychological reaction in our reviewer. We want to be dry. But I call them super mirror neurons because I think they do something that is very important. In this case, what these cells do is that something that's never been seen in the monkey recordings. That is, they increase the fire rate during execution and they decrease it during observation. In some cases, you see also the opposite pattern. Now, this is very useful. This is a very nice mechanism from which you can actually inhibit unwanted imitation. The cell fires when the patient makes the action, but it shuts itself down entirely when the patient sees somebody else making the action. It's a way of inhibiting imitation that you do not want to execute. And in the frontal lobe, we recorded from SMA proper, pre-SMA, and the dorsal and the ventral aspects of the anterior cingulate cortex. In all these sites, we have found mirror cells. They're approximately 10% of the whole cells that we recorded. And again, we're recording for about 30 minutes. That's it. Now, this is very important because the fact that you find mirror neurons also in the medial frontal cortex, of course, it tells us that the system is much more expanded in the human brain, but also speaks directly to a whole debate that it's in the mind-reading literature. Because after the discovery of mirror neurons, some people would say, well, maybe we have two ways of understanding the minds of others. One is a low-level mind reading. These are definitions from Alvin and Goldman. And in which case, these are, the mirror neuron system is a very good system for understanding the minds of others at a very low level, very simple level. But when it comes to much more complex things, you get to have a high-level mind reading, and that will be implemented in the medial wall of the frontal lobe. And there's plenty of imaging studies that suggest that if you do mentalizing tasks, you produce changes in signal in these areas. But now the fact that we have also found mirror neurons in the same corresponding areas makes me thinking that, in fact, even for high-level mind reading, mirroring mechanism at cellular level must be important. Then one day, one of my trainees came and told me, look, I found this beautiful cell, and the raster and the histogram was fantastic. And he told me, it's in the temporal lobe. How can it be? I said, Roy, it's got to be a mistake. Go back to, probably it's just a mislabeling of the site. It can't be. This cell looks so much like a motor cell. And we know that the medial temporal lobe at system level doesn't have motor properties. If you remove the medial temporal lobe to patients, they don't have motor deficits. So it took us a while to actually believe these results, but in fact we find also mirror cells in the medial temporal lobe. We have 4 recording sites: hippocampus, parahippocampal gyrus. These are 2 views. This is the coronal view, and this is the sagittal view. And the yellow circle represents the location of the electrode. So hippocampus, parahippocampal gyrus, amygdala, and also internal cortex. Of these 4 sites, these 3 contain all mirror cells. We found some also in the amygdala, but in proportion much less. This makes sense because in terms of anatomical connectivity between the medial temporal lobe and the frontal lobe, the amygdala is the region of the medial temporal lobe that is less connected to the frontal lobe. And this is an overview of the data. Probably there are many more numbers that you want to see. But the bottom line is that we have, in terms of, there are 2 major categories of mirror neurons, as I said at the beginning, the strictly congruent and the broadly congruent. And in the monkey brain, the recording shows that about 1/3 of mirror cells are strictly congruent and 2/3 are broadly congruent. We get pretty much the same thing. So about 12% of our cells, actually 11% of our cells that we recorded, have mirroring properties. And when it comes to the type of the type of action that they mirror, it's prevalent, at least in our recordings, for facial expression, but is also plenty for grasps. Overall, we have about 160 mirror cells so far. So what are the new findings? I mean, I think that it's almost trivial to say, well, we found mirror cell. When we send the paper out, Isaac wanted to emphasize this thing that we found mirror cells. Well, this is not going to, if you want to send it to this higher level journal, they're not. What is the new here? And the new is not the news. It's not the fact that we found them, but what are the new properties that we found? Well, one thing that we found was that the anatomical distribution, we have evidence for mirror cells in the medial frontal cortex and in the medial temporal cortex. So there is a large expansion compared to what we know about the monkey brain. Again, we don't know if there are mirror cells also in those regions in the monkey brain because nobody has recorded there for, in these kind of paradigms. The other interesting thing is that we found these what I call super mirrors, these cells that have opposite firing rate changes. They either increase or decrease for execution and observation in opposite ways. I think this is important for 2 reasons. One is, as I told you, inhibition of unwanted imitation. The other one is self-father distinction. If my grasping cells fire when I see Howard grasping, I have a self-father problem. How do I distinguish myself from him? Of course, proprioception is one way, but these mirror cells are also another way in which I can distinguish myself from somebody else and still having mirror cells that fire at the site of somebody else's action. But I think that in terms of general concept, I'm a motor guy, and so I always thought, well, it's all starts from action. And what mirror neurons prove is that you start from your own action, then you map the actions of other people onto your own motor repertoire. But the fact that we are also finding now these cells in the medial temporal lobe calls for a revision of the whole concept of mirror neurons, because now we have also sensory cells in the medial temporal lobe that fire for the action of the self. So again, it's a mirroring that goes both ways. And how would that work? Well, our hypothesis is just Hebbian learning. Because of the continuous imitation between people, it's very easy to associate the motor plan for an action with the side of somebody else making an action. In my seminars, I always give the example of the mother and the baby interacting. People always think of the baby imitating the mother, but it's also the other way around. There is a lot of the other way around. And so baby smiles, mother smiles back. And in baby's brain, it's very simple to associate a motor plan for smiling with the sight of somebody else smiling. And you have a mirror neuron for smiling. So just to conclude, I think this system really provides what we call a pre-reflective, because these are, very relatively simple mechanisms, just medial temporal cortex and frontal lobe, simulation-based mechanism of perception of the actions of others. Of course, it's very important for imitation. Our studies have shown that. But it's also a very important mechanism for understanding the minds of others through this embodied kind of simulation of the actions of others. But now we have also these findings in the medial frontal cortex that tell us that maybe even high-level mirroring, mind reading, even what we call reflective understanding of the minds of others, may be using some mirroring mechanism at cellular level because of the findings of the mirror cells in this medial frontal cortex. And I think that's the last outrageous statement, and I'll show you again the cover of the book. Thank you.
Thank you so much for that, Marco. I think we have time for a few questions. You ready to take questions? Yeah, okay. Looks like this one right there.
Marco, actually, I was... No? There we go. So actually, while you were talking, I was sitting here thinking, because I just had to teach this today in my undergraduate class, I was thinking, so when you started with superior temporal sulcus, I started thinking, well, what about the basal temporal language area? They've also found in epileptic patients. And so when you actually got to the medial temporal lobe stuff, I went, aha, there it is. Because, as you probably know, you get disruption of language function when they stimulate the perihippocampal gyrus with subdural electrodes. And mostly it's in the fusiform gyrus, but also perihippocampal. So I wonder, I hate to bring this up, but do you want to speculate about how, it does seem to be paralleling pretty well the organization of the language circuitry in cortex with the frontal temporal, and then you got the base of the temporal lobe going on as well.
I'm happy to speculate. I've made statements that, even just looking at the anatomy of the imitation circuit that we describe with our studies and classical language areas, there is so much overlap. And for all the other considerations, and last week there was Stephen Wilson on campus who has done beautiful work on speech perception. It seems to me that, again, mirroring seems such a nice and simple mechanism that biology and evolution has created to make what we call action and perception, because for us, we have devised these kind of labels of action and perception. The more you dig into it, the more the dichotomy and the differences really blur, and it becomes just a unique entity, something that, again, it's about mirroring. One of the analogies I make in the book is that self and other are two sides of one coin. If you split a coin into sides, what you get are two worthless pieces of metal. And I would say that for language production and perception, and from action to language, you have all these continuous sharing of mechanisms that allow the understanding, the production. And again, all these things are all about social behavior. They facilitate the comprehension, the understanding of others.
Yeah, Howard.
Yeah. Would you predict...
You need a mic.
I need this. Would you predict mirror neurons to exist in Broca's area, F5? The reason I ask is that lateral premotor cortex, such as F5, is more responsive to visual input than proprioceptive input, and the opposite's true for SMA, pre-SMA. And as proprioception allows one to distinguish self-action from observation, I'm wondering if that is somehow connected with the supermirror neuron property.
Yeah, it's a great question. I would say that I may not exclude that there are some supermirrors even in Broca's area in the lateral wall, but I would say that I bet that if you have the ability to actually map all the properties of all the cells, you're going to find many more supermirror neurons in the medial frontal cortex than in the lateral one.
Are there any regions where people have looked for mirror neurons and not found them? And the other question is, are there any physiological distinctions of these neurons? Are they just pyramidal cells? Or?
Well, these are just cells that fire. The way you define them is because you look at a firing rate change in different conditions, and that's the way you call them. That's why you call them mirror neurons. Mirror neurons, the question is, is there anybody has tried to find mirror neurons in some brain regions and didn't find them? That's a good question because, in fact, I mean, at least, again, this is such a unique situation and setup that it's not easy to replicate. But monkey neurophysiology labs are all over the world. But that's more a sociology of science kind of question. Neurophysiology, in monkey neurophysiology, is a very conservative business. In fact, I'm glad I'm not a monkey neurophysiologist because I sometimes review papers from monkey neurophysiologists for journals that also want to look at the broader impact of the manuscript. And I read the reviews of my fellow neurophysiologists, and whoa, I'm glad I'm not in that business. So it's when they find an area and a paradigm that they like, they really don't try out various things. I mean, with brain imaging, we can do all sorts of things, but they try to do what they know best. And in this case, the only lab that does it is basically Rizzolatti's lab. One thing we know is that there are serendipitous observations of mirror cells in other labs. For instance, Roger Lemon, who's a monkey neurophysiologist who studies really the motor control grasping and records mostly from F1, primary motor cortex, but also from F5. There was a postdoc from Rizzolatti's lab in Roger Lemon's lab, and they were doing simultaneous recording in F5 and in F1. And they found a mirror cell in F5 because she was actually testing for those properties. And Lemon said, well, that's very impressive. But you're not going to find tons of labs that they're going to get converted and do all mirror work because, after all, they're busy doing other things.
Can I ask a quick question?
Sure.
This question occurred to me because someone was looking at bird brains and found, as it were, mirror neurons there. So the question that they had to ask was, how do you know that when you get that response and the bird is singing, that it isn't the response to its own auditory song? So my question to you is, when you're recording from the monkey cells, the monkey, of course, is seeing its own movement as well as making the movement. So how much of the response that you're seeing is actually owed to the visual input of seeing its own response as opposed to the motor output itself? I mean, has that been controlled for by blindfolding the critter?
Excellent question. Yeah, I mean, you can actually train the monkey to grasp in the dark, and these cells fire also for grasping in the dark.
Okay, all right. So we know that it really is motor. Okay, yeah. Paula.
Yeah. Did you say that your control was like reading? Sorry, did you say that your control was reading the word smile?
Yeah, that was because for the execution part in the experiment with epileptic patients, we were showing the word smile or frown or hand or finger. We also ran a block of trials in which we show the same words and we tell our patients, now you don't move.
Right, but wasn't there early PET work showing that you could get this similar activation for making a movement or just thinking about making it?
Yeah, that's another excellent question. We found 11 cells that, in fact, also respond to the control task. They also respond just to the sight of the word.
Right, because how do you read the word without thinking about it?
And so we actually call these cells concept cells, because they seem to fire for the concept of smiling. But because they also fire for the control conditions, they are excluded from our mirror cells. We don't call them mirror cells.
Very well. I guess we can have time for one more question. Yeah. Yeah, go ahead.
Okay. I have two questions. One's a more of a technical question, and that's, do you have any estimates of what layer you're actually recording from? And the second question was an observation about one of the movies, and I was wondering on your comment, and that was the last movie you played, the monkey cell fired when the human put something to its mouth. The cell fired when it grabbed from the human, but it didn't fire when it put it into its own mouth.
Because the grasp had already occurred.
So, but I guess that's my question was, the mirroring part of it didn't seem to mirror the action of the human, but it...
But that's, in fact, it's an important concept to keep in mind, because again, if it was only strict mirroring, I don't think you would have a lot of adaptive advantage. You get to code the actions of others, especially complementary actions. We do a lot of things that are not just imitative, but complementary, that reach the same goal. And it seems that this system really seems to be coding specialized for actions and for the side of actions of others, but need not be necessarily for completely strictly congruent actions. But there are some cells that fire only for congruent actions. What was the first question? Oh, the layer. Well, in humans, we have no idea. But again, I would have a good, I would say a good guess. In the monkey, we have better ideas. And the answer is actually, if they're neuroscience students, I would ask them, where would you put mirror cells? Would you put them in layer 5, which is an output layer? You don't want to do that, because then you would really imitate it all the time. They're located mostly in the layer 3 and layer 2.